All right, welcome to our second week of Science for Healthcare Workers. Our topic for today is DNA and genetics, which is a very fascinating topic. I really enjoy this topic. So yeah, let's look at our warm-up questions. Here's our warm-up questions. Our first ones, do you look more like your mom or your dad? And do you have any brothers or sisters? What is similar about you and your siblings, and what is different about you and your siblings? All right, welcome back. I'm curious to hear what we talked about. What were some of the things that you mentioned? And specifically, what I'm looking for are kind of those traits, right? So, like, if you say, man, I look more like my mom, but why? Is it the hair color, the hair texture, your eyes, your eye color, your eye shape, your facial features, your nose? What about you physically looks like one of your two parents or your siblings? Who would like to share?
I feel like
Um,
I look more like my mom because of the hair color and eye color, and unfortunately, I'm starting to get kind of heavier in weight like her as well. But
Hmm.
yeah, and I have
Okay.
one brother, but he has more of like a square face, and I have more of a round face like mom.
Okay. Does your dad have more of a square face? Do you think your brother looks more like your dad in that way?
I don't think so, but everyone else says they look alike.
Okay. Okay. All right. Any others? Thank you.
I look like my mother, I
Okay.
think.
Okay. Why?
Um, I
What?
got the same, I got the same colors, eye color, skin color, and hair color as her.
Okay.
And
Well, I have pictures of my family for us to analyze. So here's a picture of somebody in my family. Who do you think this is?
is that your mother?
It is my mother. Some person said it
Cool.
was, some person thought it was my sister, and I told her, and she got really happy. She was like, "What?" And so, yeah, that is my mother.
She's nice
And I mean, you
looking.
can kind of see. Oh, thank you. I'll tell her that. She'll be happy. But do I look similar to my mom? What about me looks similar to my mom? I mean, it's kind of hard because it's a small photo and I'm on video too. But what do you see that's similar, possibly?
I bet you have her face shape.
Yes, we do kind of have these like same cheeks. Like you can see the kind of like
The
these
chin.
cheek shapes. Yeah. We also both have blue eyes. My dad has green eyes. My mom has blue eyes, and I have more blue eyes. So I definitely have more of my mom's eyes.
And you're
And
probably
you
as
also,
tall as her.
ooh, I am taller than my mom. Yep, I'm
Whoa,
definitely taller
cool.
than my mom. But that's kind of hard to tell on camera too. Um, For hair, we have similar hair color. I know you can't really tell. You can see all my lovely hair, right? hmm All my lovely hair that's disappearing. I definitely have more blonde hair, or I had more blonde hair. Oh, that's so sad to say. I had a lot more blonde hair when I was younger. Um, and my mom has blonde hair too. My dad has more blacker hair, and he is not balding as quickly as I am. I actually have less hair than my dad does, which is really sad. But I feel I get this actually from my mom's side because my grandfather, my mom's father, was completely bald by like 25. And so I'm not completely bald. Like I have some hair, right? I have some hair and I'm in my 30s. So I feel I'm doing okay. I'm doing okay. All right. Who's this? Take a guess at it.
That's your
It's
father.
not me. No,
No?
that's my brother. Ha!
Oh, your
I'm going to
brother.
tell him that. It's going to be great. I'm like, yeah, they say you look like my father. Nope, that's my brother. He's my older brother. His name is Jimmy. He's 6 years older than me. What do you see that's similar between us?
Nose.
Okay, our nose. Let me get a little closer.
The eyebrows.
Okay. We both have awesome beards, right? Okay, similar mouth. All right, and here's kind of a family picture. So I have my brother. This is my brother again after he had shaved his beard. This is my mom, and then my niece, which is my brother's daughter, my brother's oldest daughter. So we see 3 generations, right? We see grandma, father, and daughter. What do we see? Similar, different?
Your niece looks like you. She has your smile.
Oh, okay.
Yeah, similar smile.
Similar smile. And so that's kind of what we're talking about today. I'm going to mute everyone to keep our video nice and clean. Someone said similar hair color to her dad. Yep, my niece definitely has similar hair color to my dad, or to her dad. So when we're talking about genetics and DNA, all of that information about all of our traits, whether it's our hair color, our eye color, our face shape, nose shape, all of that information is put in this code called DNA. And that's what we're really going to focus on today is what is DNA? What does DNA stand for? What is the shape of DNA? How much different is my DNA to someone else's? Where do I get my DNA? And where is my DNA located? All right, welcome back, everyone. Perfect. All right, so what we're going to do now is there's a video that I have that's going to answer all of these questions. So we're going to watch the video and then we're going to go back and discuss these 5 questions. So here is our summary on DNA. Let me make sure we're sharing my sound. All right, and we're ready to go.
What is DNA? Every living thing has DNA, from plants to animals to bacteria, even to viruses which were once considered non-living. And most importantly, we humans have it. DNA stands for deoxyribonucleic acid, and although every living thing has DNA, its varied compositions differentiate you from others. The interesting fact is that humans' DNA is 99% the same. Therefore, it is only 1% of DNA that makes you unique. Does this mean twins don't have exactly the same DNA? No, they don't, unless they're monozygotic or identical twins. So now the questions arise. Why every living thing has DNA? What is so important in it? And what does it do? DNA has a recipe for synthesizing proteins. DNA synthesizes RNA, or ribonucleic acid, by a process called transcription that is further translated into proteins by a process termed as translation. The explanation of DNA conversion into RNA and finally RNA into proteins is called central dogma. DNA contains hereditary information that is passed on to you from your parents. If you and your father have the same colour of eyes, or your hair are as silky as your mother, it is because of DNA that you inherited from your parents. Even many diseases are passed on from generation to generation because of DNA. Where does DNA reside in your body? Your body is made up of billions and trillions of cells, and almost all cells have DNA in their nucleus. Can you tell us which cell may not contain DNA? Now let's talk about the structure of DNA. Think of DNA structure as a ladder whose rungs are made up of different bases and sides as sugar phosphate. Each composition of this nitrogenous base, pentose sugar, and phosphate group is called nucleotide. The nitrogenous bases are of 4 different types: adenine, guanine, cytosine, and thymine. Adenine will always bind with thymine, and cytosine with guanine. This type of bonding is hydrogen bonding. Phosphate and sugar are linked together to make backbone, and complementary bases are attached on it. Due to the bond angle of the sugar-phosphate molecules, the linkages will eventually form a double helix structure. There's a lot more to know about DNA, but that's all for now.
All right, so looking at our questions, we did see that word. Well, did anyone catch the word for what DNA actually stands for? It's a really long scientific word. It was here.
Deoxyribonucleic acid.
We got it. Yep. And that's what DNA stands for. All right. What is the shape of DNA? How did he describe the shape?
It's a double helix.
Double helix. Very good. And how much different is our DNA compared to other human beings?
It's very similar.
It's very, very similar. It's 99% similar.
1%.
So that means that 1% of difference, right? That 1% of difference, that 1% of code is what makes up all of those differences that we're seeing. Also, all of the genetic diseases, the genetic disorders, all of that is in just that 1% difference in the code. And so scientists are really studying genetics and DNA and looking at these really, really small differences and how that small difference can have a huge impact on a person's health and development. How do we get our DNA?
Our parents.
Right? Do you get more from one parent or the other? Or you get
I think
exactly?
It's exactly
Half
the
is
same.
from your mom, half is from your dad.
Yes, 50% from your dad and 50% from your mom. But it's a little more complicated than that, right? Because out of that 50%, right? So your mom has all of her DNA. It's not just 50% though. What part of that 50% goes into you? And what part of that 50% comes from your dad? And that's why you can have multiple siblings and all of your siblings look so different, because when we create gametes or sex cells, those chromosomes will actually swap sections of our DNA and create these different genetic outcomes for when you get half of your DNA from your mom. We don't specifically know yet what half you're getting, right? Because it's kind of a mix of all of that 100%, you're getting 50% of it. So when you have a sibling, they got 50% from your mom, but they may have gotten different parts from your mom. Does that make sense?
Does this apply to characteristics as well?
Yes.
So in the
Yep.
DNA? Yes, that could be
Yes.
that.
Yep. Mm-hmm. Yep. The only thing this doesn't account for are environmental factors. Like, for example, a big debate is like weight. How much of your body weight is genetic? You actually, like, your DNA code says this is how much your body is going to weigh, what type of a body build you're going to have, or how much of your body weight is your environment, right? The amount you exercise, the calories that you're eating, all of the things in your environment, habits like smoking, tobacco, drug usage. How much is your environment and those physical choices affecting your body's appearance, and how much of it is actually DNA? There's these two factors called nature and nurture, right? We have our DNA coming from our parents, but we also have our environment that we're growing up in. And these two forces are competing to make us who we are. That's why there's a lot of, I heard in one of the breakout sessions, people talking about twins. Twins do have the same genetic code, the same DNA, but yet they're still two different people, right? They have different personalities. They have different hobbies, different interests. And because all of those factors then would be environmental factors, how they were raised, the environment they grew up in. All right, any questions about this so far from a beginning standpoint? Any questions? Hearing none, we're going to move on. The one thing that I really want us to take away are these 4 letters, that DNA is a code. And when scientists study DNA, we look at it as a string of code, and we use these letters to represent the code. We have the 4 letters, and we know that they actually are pairs. So A will always connect with T, its counterpart will always be T, C and G always connect, and those will be the counterparts too. And here's what they look like. And they're literally like puzzle pieces, like we saw, like they actually connect and they fit together with that hydrogen bond. So that A will connect to the T and the G will connect to the C. One of the activities that I have this week for you to do is there's actually a picture of a strip of DNA and they give you half of it. You need to then create its connecting pairs. So if it's A on one side, we know its connector is going to be T. If it's T on one side, we know the other connector is going to be A. If it's C, it's going to be G. And if it's G, it's going to be C. And you're going to create a strip of DNA like we see here. That'll be one of our activities that you will do. So let's practice that. If we had our code of DNA, what would be the matching half that is connected to it? If we have an A here, what would be on the other side of that DNA, on that double helix? We have A, what would be the
T.
connector? T, very good. So we have a T, what would be on the other side?
A.
Got it. G,
And C.
right, our next one.
GC.
Next one.
G. A.
A. T.
G.
And G, perfect. And so when scientists are looking at DNA, it's easier for them to actually just look at one side of it. Because if we know one side, if we know the one half, we can automatically know what the other half is too. So we can automatically cut all of the information by just saying we're going to look at one side of the DNA because we already then know what the other side is. So when scientists are looking at DNA code, it often looks like this. They've taken one side of it, mapped it all the way out, and we then know what the other side is. And that where we can then study and look for those differences. And we can figure out what is possibly causing things like autism. What are things that are causing Down syndrome? What are things that are causing these illnesses and diseases? Are they genetic or are they environmental? Is it a virus from the environment or is it something actually in our genetics? Are some people more susceptible to certain viruses? That's kind of the debate that they're talking right now. Like, did the coronavirus affect certain types of people? And is that linked to their DNA? Is there a certain aspect of their DNA, a code that makes them more susceptible to contracting the coronavirus? Is it a specific type of blood type? And so they're mapping that information. And we've made a lot of discoveries in genetics in the last 15, 20 years. This is a very newer study, a newer science that a lot of people are making huge discoveries on. And we're going to look at some of the really cool things they're doing with DNA research. Here's some vocabulary that I wanted us to talk about. We've been talking about DNA, but we hear all these other words: genome, gene, RNA, double helix, chromosome. What do these words mean? Do we know? Any guesses? What do we got? Let's discuss some of these words. We already know DNA. We've talked about it. I want to talk about its counterpart first, RNA. What is RNA? Do we know?
No guess.
No guess?
No guess.
All right.
Ribonucleic acid.
Yes. Very good. And what is it, right? What does that mean? Do we know?
I don't remember. I just remember the name.
Or just remember the name. All right, so here are pictures, DNA and RNA. So our body makes proteins. That's what cells do. And DNA is our code, our map that specifically says what proteins need to be made. So when I think about DNA, I think of DNA as our entire code, the entire list, right? This huge list of information. And in that list is all these small pieces of information that we need. Now, when your body needs to create a specific protein, it uses RNA to do that. And RNA is basically like a shorter copy of your code. So you have your whole DNA, your RNA is just going to be a small copy of it, right? It says, oh, we need to make this specific protein. We're going to make a little copy. And notice that our copy is only one line, right? It's not a double helix. It's just one line because you only need half of the letters because we know what the other letter is going to be since they connect. So your RNA is going to be just one strip, and it's going to tell the body, this is the specific protein you need to make. And then it goes and it makes it. We're going to watch a video how this works. We're going to look at from DNA to protein and how that works.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins.
All right, so
called
there's our
histones.
entire code, right? That entire strip of DNA is the entire code of all of the information. Now, when we need to make a protein, we don't need all the information. We just need the specific information for that protein. And that's where RNA comes in. RNA is going to be just that piece of information that we need. So let's watch how that happens.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus.
So it basically takes your DNA and it says, all right, make a copy of this code. And that's what RNA is. RNA is not DNA. RNA is a little different. RNA, the way I see it, RNA is kind of a copy of a specific section of your DNA so that your body can then make a protein from it. So right now you're seeing it's making a copy. It took the DNA and said, all right, copy this section because this section is going to make the protein we need. So it's making the copy.
The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription.
All right. So now we have that copy, which is the RNA, which is not DNA, right? We copied the DNA in this shorter, smaller strip. It's a lot smaller than DNA because DNA is the whole code. RNA is that copy. We have it. That copy then is now going to was what makes our protein.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
And that's then how a protein is made. So that's the difference between DNA and RNA. They are different things, but you start with the DNA, the entire list. RNA is that little copy, that code, that section that we need to use to make that specific protein. That then goes off, it leaves the nucleus, goes into the cell body, and then the protein is made. All right, here were other words that we heard. we heard gene, we heard genome, and we heard chromosome. What are these words?
Gene is the it that decides if it's a female or male.
I don't know if I completely, kind of. I don't think I know enough to say if that's true or not.
Mm,
A gene itself. A gene is similar to a trait, like what we were talking about. So like eye color. There's a gene in your DNA. If we looked at your entire strip of DNA, there is a section of it that would be like, this is eye color. This part of your code tells you what your eye color should be. And then depending on what pattern, what code you have, what letters you have, it'll be blue, it'll be green, it'll be brown, it'll be bluish green. All the various different combinations that are out there will be in that section, that gene. So a gene is a section of your DNA that says a specific trait of what happens. All right, so then we have the word genome. Genome is almost the same thing as saying DNA, right? Genome is your entire code, right? It's your entire description of who you are. This is your genome. So your DNA is your genome. The only difference is everyone has DNA, but we all have a different genome. Does that make sense? We all have DNA. DNA is the structure. Genome is the order in which yours is structured. It's your combination of letters is your genome. We all have a different genome, but we all have DNA. Does that make sense?
Yes.
And then chromosomes. What are chromosomes?
No guess.
No guess on that one?
It's like the strand of DNA that is, or like, yeah, it's what is in your DNA to like with your genes or something like that.
Okay. I'm gonna go back to this video and look for a picture here because it showed us here when it zoomed in on this cell. So we're on the outside of the cell, and if we zoom in to the parts of the cell, we'll see the nucleus. And so here's our nucleus. And if we go inside the nucleus, I always say the DNA is inside the nucleus, but the DNA is actually organized in different chromosomes. So it's kind of chromosome is the shape, and then DNA is the smaller parts of it. So let's take a look. We're going to get a little closer here. So if I go inside and zoom into that nucleus, we'll see that the nucleus is made up of these different, and they kind of look like X's, right? They look like little X's, and those are our chromosomes. And we have various chromosomes. Do you know how many chromosomes we have in the human body? We have 46 chromosomes in our body. And so looking at these chromosomes, this like X shape, that is your DNA, but it's like folded and wrapped and folded and folded and folded. And your DNA kind of floats around in this X shape inside your nucleus. And that X shape is your chromosome. And your body inside every your nucleus will have 46 of these X's of your DNA folded and going, because this blue line is that strip of DNA, and it goes back and back and back and back and back and back, back and back and back, because your DNA is really, 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 really long, and it's going back and forth, almost like a coil or like rope that you've just like stacked up, or Christmas lights that just go back and back and back, and it's all just kind of tangled up, going back and forth. Again, making it difficult for scientists to study, right? Because you have to then like untangle the whole thing to then see what the specific code is and in what order. So there's our chromosomes. So those are all of our, any questions about these vocabulary words? Are there any of them that are still not clear? All right, so going back to this process of making proteins, right? So we had the DNA, the RNA was that copy of what we need to make a specific protein, and then we made that protein. Why is that process important, and how is it related to viruses? Last week, we kind of talked a little bit after class about the coronavirus, right? And we were talking about the vaccine and how does the vaccine work? And the vaccine for the coronavirus is part of this process. Because, so here's an image. We see a virus, a corona-like virus, and we see the cell membrane that we had read about. And the way that viruses work is that a virus will attach itself to the cell. And then the DNA, we see the virus has its own DNA. Now, scientists kind of argue, there's a debate in science right now on whether viruses are living organisms or not. A lot of people argue viruses are not living organisms because the viruses are not able to replicate. They cannot beget. They cannot make babies, right? All living organisms can reproduce, right? That we can get together with our pairs, we can reproduce and make babies. Plants do the same thing, right? They produce pollen, they produce seeds, and then they reproduce. Viruses cannot reproduce. And if viruses can't reproduce, how do they spread?
They hijack other cells?
Yes. So our cells are really good at copying DNA, right? I mean, you saw how we had DNA and then it creates RNA, and our cells are really, really good at reproducing and copying DNA. We're constantly copying DNA and making new cells because our cell will make 2 cells, which will make 4 cells. We're constantly dividing our cells because that's how we grow. We grow by taking one cell and dividing it. And when that happens, our DNA makes a copy of itself when we have cell division. So what viruses do, like here in this picture, it will attach itself to a cell. It will take its DNA and it will insert it into a host cell. And then it tricks your host cell into thinking, this is your DNA. This is your DNA now. So the cell takes it and say, oh, this is our DNA. And it starts copying it. And it copies it, and it copies it, and it copies it, and it starts making the virus, basically, because that's what the virus is its DNA. So our cells make copies, making more and more and more of those viruses. That cell then eventually dies, and now there's multiple copies of that virus that go out and affect more cells that then die, and then it goes out and affects more cells, and then goes out and affects more cells, and then eventually it leaves the human body looking for another human body with cells to infect. And that's how they grow. That's how all viruses work. They use its host cell to copy its DNA because
So it's
it
a
doesn't.
parasite.
Yeah, it's basically like a parasite. Yep. It lives off of another organism. The only difference between a parasite, though, is parasites can still perform reproduction. Like if you think about like the worms and the different parasites, they can still make their own babies, right? They lay eggs, they produce babies, but parasites need another host to live, right? They're getting their food from another living organism. They've attached on and they're sucking the blood, they're living off of it, but then they're still able to make their own babies. What's different about a parasite and a virus needs another living organism for reproduction. Does that make sense?
Yes, it does.
Does that make sense? Perfect. So that's how viruses work. So now we're talking about the vaccine. Well, how does a vaccine work? What we are trying to do with the vaccine is create a protein that stops the coronavirus from attaching to host cells. We're creating a protein that will hit these little like connectors and actually stop and prohibit the virus from attaching to cells, and that means it'll stop the DNA from entering the cell as well. So that's one way, one type of vaccine is working, is creating this protein. But the question is, how are we going to do that? How can we get our body to create the protein that we need to attach and stop the virus from attaching to the cell? Well, there are 3 steps to the process, right? We have the DNA, which is our code that tells us what proteins to make. We have the RNA, which is the copy of the code that says make this protein. And then there's the actual protein itself. When we create a vaccine to tell the human body to create proteins to protect itself, how are we going to do it? Are we going to insert it into the DNA? Are we going to give it RNA to then just make copies of the protein? Or are we just going to dump a bunch of proteins into the body? Which one are we going to do?
RNA.
Yeah, there are many options. And so the one right now that the vaccine, like the Moderna and the Pfizer vaccine are, if you read them, they're called mRNA vaccines, because that's what it is. We're giving the body that code to say, hey, make this protein. So we insert it into the body reacts to it just like it does its own RNA and protein. So the body starts making that protein, and then those proteins attach to the virus, stopping it from reproducing inside the body. How cool is that? But how do we do it? So there are different types of vaccines. One of them is DNA and RNA vaccines, where you literally tell the body, this is the protein that you need to make. Other vaccines that we have, there are some where they've used an actual like live version of the virus, but it is a weakened form of the virus. By giving you the actual virus itself, that allows the body to do its natural process to say, ooh, there's this invader. What do we need to do? The body eventually finds out, well, we need this protein to stop it from connecting to us. So quick, make this protein. And then it inserts it into your DNA. It creates the RNA, and then those proteins are made. Another way that they can make vaccines are also using inactive viruses, which means they basically create something that looks like the virus to your body. When your body sees it, your body says, oh, this is the SARS-CoV virus. I need to create this protein. But it doesn't actually have the DNA and some of the structure, so it's not going to reproduce, it's not going to behave or harm you like the virus. It is just a, what's the word? I can't even say clone, right? It's just, it looks like it, but it's really not the virus. It's like a fake, a placebo. It's supposed to look like it. So your body reacts naturally. The current vaccines that we have right now that we're looking at, there are many different types of vaccines that they're working on. The two that are being distributed right now are RNA vaccines. And that is where we're telling the body what protein it needs to make. Does that make sense?
Yes.
As we see that, the mRNA COVID vaccine. All right, so I wanted to talk a little bit about the future of what we're capable of doing in the near future. So we have, scientists have developed this tactic called CRISPR, CRISPR-9, Case9, CRISPR Case9 is what it's called. And we're thinking, can we actually attack the DNA of viruses? Can we actually send something to say, when the DNA of a virus enters your body, is there a way that we can attack the DNA saying, nope, we're not going to make copies of you because you're bad. You're not actually our DNA. And we've identified that you're not our DNA. Get rid of you. And are there ways that we can change our own DNA so that we can edit the human genome? Are there ways that we can change mutations, that we can fix inaccurate code or code that's causing things like Down syndrome, sickle cell anemia, all those genetic disorders? Are there a way that we could just clip the DNA, change it, and put it back together? Can we edit human genome? And we can. We're starting to discover that we can do that. And so here is the future of DNA research and the creation of potential vaccines of how we can fight viruses. Let's watch. This is cool stuff. Oh.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA.
Now,
one of
this
which contains a
process
sequence
actually
that
happens
matches that
right
of
now.
the
There are
invading
some
virus.
bacteria, and that's why it's called CRISPR. They actually found it in a type of yogurt because there was a yogurt, right? And there's bacteria in yogurt, and companies who create foods like yogurt, cheeses, they're very they're invested in keeping their bacteria alive, right? Because that bacteria is what produces the yogurt and the cheeses. And so those companies were investing a lot of technology into figuring out how can we stop viruses from affecting our bacteria that's creating our products like yogurts. And what they found is that there were certain bacteria in nature that had this CRISPR-Cas9 capability, that when a virus would enter the bacteria actually just destroyed the DNA of the virus, not reproducing the virus inside of it. And scientists went, wow, this is amazing. Could we actually do that inside the human body? Could we teach human cells to do the same thing that these bacteria cells are doing in yogurt? And watch this.
These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA.
So it has that
When the matching
RNA, which is
sequence,
that
known as
guide
the guide
RNA. So
RNA,
it's telling this protein saying, hey, look for this strip of DNA. Because if it has this strip of DNA, it is not our DNA, it is the virus's DNA, and you need to cut it and destroy it. So it is going to start scanning every bit of DNA inside that living organism. And if it finds that strip of code, it's going to say, oh, this is the virus. Let's snip it.
finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic
So that is
disease.
the future of genetic science. Thoughts? What do you think?
That will be amazing.
Mm-hmm. Where we could start changing the DNA and changing kind of those bits of codes that are saying, oh, you know, sickle cell anemia, all those mutations, why we have genetic disorders and genetic diseases, we could technically edit them out. What are your thoughts on that? Should we do something like that? Should we not? Also, the big debate is, where do we stop? Once we start actually changing the human genome and say, oh, we're going to delete and change this part of the DNA. Oh, we're going to delete, change this part of the code. Oh, delete, change this part of the code. Where do we stop? Oh, we could make people smarter. We could make people stronger. We could make people taller. We could make people need 6 hours of sleep, not 8 hours of sleep. We could start programming our children to be exactly what we wanted them to be.
And then will we still be people after that?
Mm-hmm. Yeah, that's the question. At what far is too far, right? And that's where science is having this debate on ethics right now. We have the technology to make changes and edit the human DNA, the human genome, but should we? Should we cure sickle cell anemia? Should we cure Down syndrome? Should we cure some of these genetic mutations? Should we not? Where's that line of too far? But yeah, this is the future of vaccine research, working with viruses, and also just human development and growth. All right, so we're going to step away from DNA a little bit and look at genetics itself and hereditary and how we get our stuff. So we've already talked about our DNA, that we have 46 of these chromosomes, these little X's, which is all of our DNA folded on top of itself. And we get 23 of them from our mom and we get 23 of them from our dad. But the question is we don't know which 23 we're getting from your mom. And we also don't know which 23 we're getting from our dad. So it can be a mix. And that's why all of your brothers and sisters look so different from you, because you got some from your mom and you got some from your dad, and they got some from your mom, but they got different ones. Some are the same, some are different, some are same and some are different from your dad. And they get all of these to make your total of 46 chromosomes. So looking at genes, right? So we have our genes, which are these like sections of code. And then there's always a pair, right? There's always a matching pair on the other side. So there's two parts. And that's when we look in genetics, there's always kind of two things. And like with chromosomes, we talk about Y and X chromosomes, that there's always these like pairs and combinations. And when we're looking in genetics, every trait, every gene has two possibilities, and they're combining those two together. So you have half of the chromosomes from your dad and the other half, right? Because you're getting 50-50. You have 50% from your dad, 50% from your dad, and you don't know which one of those two you're going to get. And same thing from your mom. You have 50% from your mom and another 50%, but you don't know what 50% you're getting from your mom. So the way that these combine, we have 4 different outcomes when we're looking at genetics. So when we look at every genetic trait, you can have 4 different outcomes, and you are going to be one of those 4 different outcomes. Let's take a better look. So out of genes. Another vocabulary word you will hear is alleles, and alleles is just the same thing for trait, right? So we have hair color, we have eye color, we have hair length, and all of that are these like small sections inside your DNA called alleles. And depending on what letters you have, A, T, C, or G, that will say blue eyes, brown eyes, green eyes, etc. So let's take a look. So let's say your dad has greenish blue eyes. So the way we code that, we can give him what's called a genotype. And we're going to say, since he has greenish blue eyes, we're going to give him a big B and a little b, and that together makes his genotype. bluish green eyes. But when he has a child, those are going to separate, and he could give the capital B or he could give the little b to his child. We don't know which one of these two he's going to pass on. He has the ability to pass on either one of these, but only one of them to his child. So our mom has blue eyes, and since her eyes are completely blue, she's going to have two capital B's. So capital B, and one of those two is going to be passed on to the child. And so we have four different outcomes. We can get the blue genotype from the mom, and we can get the blue genotype from the dad, which means this child would have blue eyes. We could get the blue from the mom here, and we could get the blue from the dad, which means this possibility would be blue eyes. We could get the blue, and we could get that bluish green variant, right? That lowercase. And then same thing here, we could get the blue from the mom, and we can get that lower blue case variant from the dad. So that gives us four different outcomes that the children, the offspring, could have. And so looking at this, the way that it works out, 2 out of the 4 are going to have blue eyes, and 2 out of the 4 are going to have greenish blue eyes. And that's why, again, when you look at all of your siblings, your brothers and sisters, why you have all of these variations, because half is coming from your dad and half is coming from your mom, but you don't know which half is coming from your dad, and you don't know which half is coming from your mom. So there's always going to be 4 different possibilities. Let's take another look here. So my brother has greenish blue eyes because he got a big B from my mom. My mom has blue eyes and my dad has green eyes. So my brother had, he got blue eyes from my mom and green eyes from my dad. So he has the big B and the little b. Now his wife that he married also has the same colored eyes. That kind of greenish blue, that hazel colored eyes, because one parent had blue eyes, one parent had green eyes, and had kind of gotten this mixed. So they have these genotypes. If I look at the possibilities, the dad could pass on the capital B, and the mom could pass on the capital B, which means this child here would have solid blue eyes. Even though both parents kind of had this like hazily colored eyes, one child could have solid blue eyes. Here, if we got the capital B from the mom and the lowercase b here, this child would have that greenish blue eyes because it has both. If we look here, same thing, we have a capital B from the dad, that lowercase b from the mom. So this child would have that mix, that hazel blue eyes. But one of the child could actually get the 2 lowercase b's from each parent, lowercase from the dad, lowercase from the mom, and this child would actually have green eyes. So if we have a parent, a father with greenish blue eyes and a mother with greenish blue eyes, in theory, if they had 4 children, one of them could have blue eyes, one of them could have green eyes, and 2 of them are going to have that kind of mix hazel colored eyes. Does that make sense?
Yes, it does.
Yeah.
Yes.
And that's why we get all of those different variations that we see in our family. So here's some vocabulary words. We talk about genotype, and genotype is our specific code that we have. Is it blue? Is it the code for blue? Is it the code for green? Is it the code for hazel green? Is it whatever color? Is it the code for brown? That is your genotype, is the specific code that you have. Another word you hear is phenotype, and that's the description. What do we actually see? I see blue. I see green. I see bluish green. These are words that we use a lot in genetics, because sometimes you can actually have someone that has like a trait in their actual code in their DNA, but you don't see it. And that's where we get into things called recessive traits, which makes this is where genetics starts getting really complicated. So recessive traits are, you have a code in your genotype that you can pass on to your children, but you don't necessarily see it. So for example, if someone has two capital B's, they could have brown eyes. If they have a capital B and a lowercase b, they're going to still have brown eyes, because brown is a dominant trait. If there is that brown trait somewhere in the code, it will be brown, because brown wants to dominate. Blue is a recessive trait. And so for blue, that child has to have two lowercase b's. So let's take a look how that works with dominance and recessive. So we have one person with brown eyes and we have one person with blue eyes. And if they have a child, let's say they had 4 children, how many of them could potentially have brown eyes? How many would have brown eyes? What's the possibilities of having brown eyes?
50-50.
You think it's 50-50? Any other guesses?
3.
3 out of 4? So
Yeah.
three will have brown and only one
One
would
blue.
have blue eyes.
Yes.
All right, let's take a look. So the capital B from the dad is going to go here, and the lowercase b is going to be from the mom. Since there is one capital B, actually, let me do this better. I'm going to type. There we go. That's so much clearer. Since there is one capital B, this will be brown. Same thing here, we're getting a capital B from the dad, that brown trait, and we're getting the blue trait from the mom. Since there's one capital B and that's a dominant trait, it's going to be brown eyes. Here, same thing, we're getting that capital B from the dad, and we're getting that lowercase from the mom. So that trait's going to be brown. Here, we're getting that capital B from the dad and that lowercase b from the brown. There's one capital B, so that one is also going to be brown. What color eyes are they going to have?
All of them are gonna have brown eyes.
All of them are going to have brown eyes, because the dad has 2 dominant traits, right? And so he's always going to pass on one of his dominant traits. All right, but let's say we have a person with brown eyes, but his mom had blue eyes. So his code is actually capital B, lowercase b. He has brown eyes, but there's people in his family with blue eyes. So that trait is in his code. It's in his DNA. He has it. He's carrying it with him, and he could possibly pass that on to his children, but you don't see it because his phenotype, what you see, is brown. So here we have the wife that has blue eyes. We know they are both lowercase because she has that recessive trait of blue. Now, how many of them are going to have brown eyes? How many are going to have blue eyes?
Half and half.
Half and half? 50-50? Any other guesses?
I say half and half again too.
You say half and half again? All right, let's take a look. So here, what am I getting from the dad? The dad up here with brown eyes, is it going to be a capital B or a lowercase b?
Capital B.
Capital B. And from the mom?
Lowercase.
Lowercase b. And so what color will we see?
Brown.
We'll see brown. Oh, I can't spell brown today. Brown. There we go. All right, next one. What are we getting from the dad?
Lowercase and lowercase.
Lowercase
Yep.
and lowercase.
It'll
So
be
what
blue.
color? It will see blue.
Yes.
All right, down here, what are we going to see? What are we getting from
Capital
the dad?
B.
Capital B. And from the mom?
Lowercase b, which is
And
another
so-
brown.
Which we'll see is another brown. I did
And
it
then
again.
this
And then here, lowercase, lowercase.
will
We're
be
gonna get
blue.
And so this child will have a 50% chance of having blue eyes or a 50% chance of having brown eyes, because brown is that dominant trait. And this is actually true. We know that brown is a dominant trait in eye color, and that's why the number of people with blue eyes is decreasing, because more and more people with brown eyes and blue eyes are choosing to mate. So if you have blue eyes, stop mating with people with brown eyes. We need to save blue colored eyes, right? Right?
It's all in love, my friend. It's all in love.
It's all in love. All right, now let's look at this one. So we have two parents, they have brown eyes, right? So he has a brown eye, but there's someone in his family with blue eyes, so it's in his DNA. He has that recessive trait. You just don't see it. It's hidden, it's a recessive trait. Same thing, he has a child with someone with the same thing, has brown eyes, but there's someone in the family with blue eyes. That recessive trait was passed on. Now what do we got? How many are going to have brown and how many are going to have blue?
3 brown, 1 blue.
All right, 3 brown, 1 blue. Let's double check it. So here this person's going to have both dominant, so that will definitely be brown. Here they're getting the lowercase from the dad and the upper, but I'm going to write the capital letter first. So again, it's getting that dominant from the mom, so it'll definitely be brown. Here they're getting that dominant B from the dad, the recessive from the mom, so again will be brown, but they could potentially have children with blue eyes. This person will not ever have a child with blue eyes because they have 2 dominant traits. They are no longer passing on the blue trait. They do not have that blue trait in their DNA code. And then this person here will actually have 2 lowercase b's. And that's where we do have situations where we have 2 parents with brown eyes and they have a baby and they're just like, oh, the baby has blue eyes. How did that happen? It can happen because of the way, as long as both parents have that blue trait in their DNA, you have a 25% chance that that will be passed on to the child. 75% will be brown and 25% will be blue.
That's crazy because from our side, we both have brown eyes. I don't know exactly if she has any other colored eyes in their genes through their side of the family, but I know for sure that my side of the family, my mom has blue eyes.
Okay.
But I came out with golden brown eyes. So.
And what color eyes does your daughter have?
My daughter has beautiful bright brown eyes right now, but still kind of changing. But my son has different colored eyes. It's weird because with us, his mom has green eyes and I have brown eyes, but the trait of blue eyes sit within me from my mom. And his eyes are like a hazel, like a grayish bluish looking.
Yeah. And what I am talking about here though is like super simplified, because in actuality, if we were to actually look at the DNA and the code of eye color, there are so many different colors of eyes, right? It's not just brown and blue and green. There's so much, and there's so many codes, right? And this is so much more complicated than just blue and brown, green, hazel. But it just kind of shows you how it works and how you can have 2 parents with very dark brown eyes, and then they have a child with light blue eyes. It is possible through the way recessive traits work. All right, let's do some readings here. We have about 5 minutes left. Our first reading, I think we're going to go here. packing. So I have a reading here, Packing DNA in Our Cells, and it talks about chromosomes and it like describes the shape of them. I would let you read this on your own. It'll be in the slides, but because of time, we're going to skip this one because we talked about the shapes of chromosomes. We're going to look at the seed color. So when we look at genetics, it's not just human beings, it's all living organisms. And we can do the same thing with plants. And plants reproduce the same way, right? 50% of the genetic code is coming from one plant and the other 50% is coming from the other plant. And then it is this swapping of DNA. Can I have someone read this paragraph about seed color in people?
The two alleles that make up a gene pair can differ. When a gene's alleles can differ, the trait produced by the gene can vary. If a dominant allele is present in an organism, the organism has the trait associated with the allele. Genotype, or the makeup of the alleles in gene pair, is represented by symbols such as YY slash YY, lowercase y, or lowercase y and lowercase y. A capital letter indicates a dominant allele. The Punnett square shows the potential genotypes for seed color in the offspring of two particular pea plants.
Excellent, very good. So let's look here. So here we see, and our capital Y means it's going to be more of a yellow and it's dominant. So if there's at least one capital, it's going to have a really yellow color. If it has a lowercase y, it has that recessive gene of it'll be green. So I see a capital Y, capital Y, capital Y, and I see two lowercase y's. So how many of these will be yellow? What's the probability that it'll be yellow?
3 to 1.
Yep, 3 to 1. Yep. So 3 to 1. 3 will be yellow and 1 will be green. All right, let's read our questions. Would someone like to read our first question?
Is yellow green in some plants? Oh, the Punnett square suggests that seed color in pea plant.
What do we have? Yellow green in some plants is determined by more than one gene, is passed on to offspring, is controlled by a gene with alleles that can differ. What do we know about seed color in pea plants?
D.
It is D.
Yeah.
Controlled by a gene with alleles that can differ. All right, and number 2. Who would like to read question number 2?
Based on the Punnett square, what is the probability of producing a plant with yellow seeds?
And we said 3 out of 1. And there was a total of 4 of them.
75%.
So what percent was?
75%. C.
Yeah. 75%. 3 out of the 4. Pretty good. All right, so that was our first reading. There's another one here about genotype and phenotype, specifically looking at freckles that you can read. This will be in the slides on the Google Classroom, and a Punnett square for you to do here to mark. We have one over here over seed texture, and the same thing, looking at different ones. And our next one is the same thing, looking at different ratios and probability of what are the potential outcomes for that genetics and heredity. So all these activities, all these readings you can do, they'll be in the slides posted on the Google Classroom. Let me hop over to the Google Classroom right now and give you a little peek at what we have for activities for this week. So let me pop over here. So when you get on our Google Classroom for week 2, I've uploaded 3 assignments. You have one assignment here that is about Punnett squares that we were just doing. It'll be a website with some vocabulary words to look at. They'll give you examples of Punnett squares and even some activities where you can actually fill out the Punnett squares themselves and look at the probable outcome of heredity, of what will be the outcome of the offspring potentially. Assignment 2 is completing the DNA that I told you about. It gives you half a strip of DNA. You need to match the letters, and then there'll be a strip of RNA for you to then match with it. There's one thing I wanted to point out. I just realized I didn't mention it. I'm glad I covered the homework. I didn't talk about this, but the actual code itself, the C, the G, and the A are all the same, but the T is not a T in RNA code. When we write RNA code, it's actually a different structure, and we use the letter U for it. So when we write strips of RNA, if there's an A, the opposite, so we actually write U instead of T. So that's something to keep in mind too. So if we're taking a strip of DNA and we're creating an RNA copy of it, every time there's a T in the DNA, we're actually going to use the letter U for the RNA, because RNA is not DNA. That's that copy of the code, and U is the only letter that is different when we write that code. So that is something you'll do in assignment number 2. And then assignment number 3 is actually kind of fun. It's called DNA for CSI. It gives 4 different examples and 4 different questions that you kind of have to act like an investigator, just like CSI, and figure out who did it based on the DNA samples that you have. And you have to cross-reference the DNAs. And the thing is, when you're looking at DNA samples and you're looking at relatives, when you have a parent and a child, a lot of their DNA will be similar, but only 50% of it, right? Because the other half, they're still getting from that other parent. So you kind of have to think about that, of which one is most likely the relative or the parent based on what is most common in the DNA. Which is kind of a really fun activity. So those are the 3 activities. I posted a really interesting YouTube video you can watch that shows how DNA and genetics affect this idea of evolution, and really talks about like that heredity, right? That as we pass on traits and we pass on genes, which ones start to disappear, which ones start to grow more, and how does that affect the evolution of a living organism and the species in the future. And then here's a link to the presentation slides. So you can always go back and relook at all of the slides, relook at the videos, and do those readings at the end. And then I do have a reading from our anatomy textbook for like the nursing program.